Hello everyone! How are you doing? Welcome to another MaxWell webinar. For all of you who have watched our old webinars, welcome back. And for the ones who's just watching our webinar for the first time, thank you so much for your interest, and I hope you enjoy today's session. So I'm just now waiting for our attendees to enter the room. Hello everyone, nice to see you. All right. So before we begin, I just wanted to introduce to you the platform that we are using now. For the entire webinar, you will see the slides and also the speaker on the upper right corner. And whenever you have some questions, you can write them on the chat on the right side. So you can ask directly our team, our webinar team, if you have any questions on the tool itself, the webinar tool. If you encounter any technical issues, just feel free to add your requests there. And also, if you have specific questions about the presentations, that's also the place to add in your questions. Another thing is for this webinar, we'll share some links relating to the paper that is featured for today. So just check out these links on the chat section. Yes. All right. So I see that people are coming in. Thank you so much. We have people from the US, from Europe, from Asia. Thank you for coming and attending our conference, our webinar for today. All right. So let's start. So for today, we will have our episode 4 of MaxWell webinar, and we have 2 guest speakers. The first one is Maria Sundberg. She's currently a research fellow at the Boston Children's Hospital under the supervision of Professor Mustafa Sahin. Our next speaker is Hannah Pinson. She's currently at the Vrije Universiteit Brussel as a PhD candidate in the group of Professor Vincent Ginis. And for the work that will be presented today, she did that while she was a visiting scientist at MIT in the group of Professor Max Tegmark. So we're very excited to welcome both Maria and Hannah. So what are we going to talk about today? First, so that's me, Marie. Nice to meet you all. I will be your host, and I'll just speak about MaxWell Biosystems and about our technology, a microelectrode array. And then we'll proceed to the scientific presentation by Maria and Hannah, of which the title is Phenotyping Neurodevelopmental and Psychiatric Disorders with Human iPSC-Derived Dopaminergic Neurons. And for today, you will see an entire overview of the project, starting from the motivation to the methods that were used, until also some focus on the results generated using the MaxWell system. At the end of the presentation, we'll have a Q&A session wherein we will go through your questions in the chat. And what will happen is I will ask the question, and then our speakers will answer them. And then if there are some questions that are not answered within the session today, we will send you answers via email. So feel free anytime to add in your questions in the chat. All right. So about MaxWell Biosystems, for those of you who already know us, I wouldn't need to explain more, but for the new ones, we are based in Switzerland. We founded a company in 2016, and it is our mission to provide electrophysiology systems and solutions to advance scientific discovery and to accelerate the development of novel drugs for brain diseases. So we're really focusing on neurons. And our core expertise in the company is to develop microelectrode array platforms, both from the hardware perspective and also the software, and also data analysis. And we are experts in the applications of such high-density microelectrode arrays, and we have more than 70 journal papers using the system. So for our customers, we have labs from academic institutes, from contract research organizations, biotech, and also pharmaceutical companies. So what are high-density microelectrode arrays? So here I'm showing an image of a neuron wherein the activity, the electrical information or the electrical signal of that neuron can be read by 2 different ways. So the typical one, which is gold standard, is by patch clamp, which you can see here as these needles close to the neurons. So this is patch 1, patch 2, patch 3, and you can see the signals that can be seen here on the right side. So these are action potentials that can be detected by attaching these glass micropipettes on different parts of the neurons. The advantage here is that you can detect such signals in high quality. Now, high-density microelectrode arrays can also detect these action potentials, but here we have multiple electrodes that are tightly spaced with each other, such that we are able to detect the signals not only from 1 or 2 or 3 spots of the neuron, but multiple spots. So we can then see the activity on the axonal area, the somatic area, and also the dendritic area, as what is shown here. So it's a very powerful tool for you to be able to understand how the activity is generated and how it propagates along single neurons. And then you can imagine all the neurons in one sample in each of the well. So what are the key advantages of high-density microelectrode arrays? So the lower resolution MEAs are very well used in the industry, in academia as well. But here, you're getting mostly the average signal of your sample. So in this figure here, you will not be able to very quickly understand whether you're recording from 1 neuron or 3 neurons or 5 neurons. There are also chances wherein you might miss some of the neurons in between the electrodes. While for high resolution, which is what we have for our technology, there's always an electrode at the right spot to capture the activity of every cell that is in the sample. Additionally, we have these small electrodes, which allows us access to the cell's small signals. That's why we're able to capture these propagating signals along the axons of each of the cell. With this, we are able to read out across different scales, from the network level, like the whole network activity, to individual cells, cell level, up to subcellular level, which is related to, for example, propagating action potentials along the axons. So what are our high-density microelectrode array platforms? We have two. The first one is called MaxWell, the second one is called Max2. So for Max1, it's a single HDMEA system wherein we have a single chip with 26,400 electrodes in total. I will tell you more about the sensor, the chip later. So we have a recording unit which is very small that can fit inside of the incubator, and then you can keep the recording your activity inside of your main incubator. For Max2, it's basically 6 or 24 wells version of our Max1, and our mainframe can be used for data acquisition, and it's also a built-in incubator. And you can have your recordings in this mainframe outside of your main incubator. So this blue door closes and it seals, and you have the temperature controlled and also gas input, and it can also be humidified. Now focusing more on the electrodes. So here in each of the well, we have 26,400 electrodes within a 2x4 millimeter square area. And the distance between these two electrodes is 17.5 micrometers center to center. So what you can imagine now is for every cell, as shown here on the figure on the right, one neuron can be detected by multiple electrodes, and also the neurites or the axons can also be passing on top of these electrodes and can be detected as well. These signals coming from those axons can be detected as well. And additionally, the reason why we can do this is because even with these small electrodes, we have low noise readouts, 2.4 microvolt RMS. And any of these 26,000 electrodes can be chosen to stimulate the neurons. So we can deliver electrical stimulation on any of these electrodes. Now, focusing on what we can do with this technology. So this is just a rough overview of our assays. You will learn more from the presentation later. So first, we have activity scan assay. Here, we are able to identify where are the cells located, how active are they, and what is the amplitude of the signal, and other features of all of your cells in the sample. So on top here, you see the fluorescence microscopy image of the same sample that has been recorded using Max2, wherein you're able to detect the spots where there are cells. So you can see kind of the same patterns, like you can see in the middle, some circular area, which is also visible in the figure below. Here what we show is the firing rate. So the reddish spots are the more active neurons, and the bluish spots are the less active neurons. Next, after identifying where the cells are, you would want to continuously record them. So in our network assay, automatically these neurons are then identified, the location, and then we are able to record them continuously for a little bit longer term. So here is a sample raster plot wherein one line corresponds to the activity of one electrode, one of these 26,000 electrodes, and each dot corresponds to this action potential generated by that by the neuron nearby that electrode. So whenever you see these bars, that means a network has already formed in the sample, and since they are interconnected, whenever there is activity, most of them kind of follow or become synchronous to the activity that is happening at that same time. So you can summarize that into this network activity below. So when you see a peak, which is just a sum of all of the action potentials during that time, that means that's the peak of the network activity, and then it goes down and you see some oscillating network activity in your sample. So of course, this feature is different depending on your cell line. And the third one here, which we are super excited about, is the axon tracking assay. Here you can really zoom in each of the cell, identify where the action potential initiates, and then how it propagates along the axons. So you're able to extract the morphology of the neuron just based on electrical activity, not needing to to add any compound or to add any dyes on your sample. And we can then use our algorithm to be able to track these branches. And for every branch, we are able to then compute the conduction velocity along these axons, which is shown here below. So as the action potential propagates, we are able to follow it, compute the velocity, and this may be a very important metric if you're, for example, comparing the excitability or the health of your neurons. All right, so that's for our quick intro about our technology. I would also like to highlight that recently we got these very cool publications using our technology. So the first one in the list is the one that will be introduced today by our speakers. So it has been published in Nature Communications, and we are super excited to hear about this work on dopaminergic neurons. Another paper that has been recently published is this one by Sylvia Ronke from Andreas Hierleman's lab. And here, basically, the main work is characterizing different cell lines, iPS cell lines, using high-density microelectrode array. So please check it out as well. And lastly, for all of you who are interested in doing organoids, there is now a bioRxiv preprint that has been up recently by Tal Scharf in Ken Kosik's lab. So now let's proceed to our main presentation. So I'd like to welcome Maria. Hi, Maria.
Hi.
Hi. So I'll now transfer the floor to you. So I'll stop my presentation.
All right.
Okay, thanks. Go ahead.
Thank you for MaxWell Biosystems for this opportunity to present our work here today. I am going to talk about our recent study of phenotyping of neurodevelopmental and psychiatric disorders with human iPS cell-derived dopaminergic neurons. Here is the outline of my presentation. First, I'm going to introduce a copy number variation 16p11.2 in the brain development, and then I'm going to tell you about characterization of dopaminergic neurons with 16p11.2 copy number variations, and finally, I will go over the methods used in this study. 16p11.2 copy number variations have been implicated to be present in several neuropsychiatric and neurodevelopmental disorders like schizophrenia, bipolar disorder, and autism. Autism is more common in patients with 16p deletion, whereas schizophrenia and bipolar disorder are more common in 16p duplication. Brain imaging studies have shown that in the patients with deletion, patients have macrocephalic brain growth, which is increased brain volume, and the patient with 16p duplication have macrocephalic brain growth, which is decreased brain volume, shown here in this picture in blue. We were studying dopaminergic neurons because previous studies have implicated that there are deficits in dopamine signaling in autism and schizophrenia. Also in autism, irritability and anger have been treated with common antipsychotic medications like haloperidol and risperidone, which are dopamine receptor blockers. In schizophrenia, psychotic disorders have been treated with these dopamine receptor blockers as well. This indicates that dopamine signaling is disturbed in these disorders, and that is why our aim was to differentiate iPS cells that carry these copy number variations 16p11.2 deletion and duplication into dopaminergic neurons to find new molecular targets for drug development for these disorders. Here is the outline of our study. First, we have human iPS cells in culture. Then we used CRISPR-Cas9 gene editing method to generate isogenic control lines and control lines that carry 16p11.2 deletion or 16p11.2 duplication. Then we differentiated these isogenic lines into dopaminergic neurons in vitro and characterized their phenotypes with measuring different aspects of them, for example, dopamine neurodifferentiation capacity, electrophysiological properties, transcriptional profiling, to identify molecular targets and test different drugs for these cells to, in the end, develop personalized medicines for these patients. Here is the data of our results. To show that the iPS cells are pluripotent, we stained them with the pluripotency markers OCT4 and Raman60, shown here in red and green. We also wanted to verify that the iPS cells contain the copy number variations. And here in the blue, we saw that 16p duplication cells have increased expression of the genes from the 16p gene region, shown with QRT-PCR method. In the 16p deletion cells, showing here in red, these genes were downregulated. Then we differentiated these iPS cells into neural precursor cells, and in the panel D, we show that these cells still retain their copy number variations. As shown with blue, the 16p duplication cells have increased expression of these markers, whereas the 16p deletion cells, showing here in red, have decreased expression of these markers. Then we differentiated these iPS cells into dopaminergic neurons by using previously published methods with small modifications. The neural induction was induced with knocking an SP drug, floor plate patterning was induced with SAG, and Wnt signaling activation was done with Kyer. The cells were sorted with FACS sorting method with NCAM positive selection at day 27 to day 32 of differentiation. This was done to enrich the dopaminergic neurons in the cell populations. In the panel B, we saw the sample time collection time points for QRT-PCR, immunocytochemistry, high-density MEA recordings, and low-density MEA recordings, and PATS-CLAMP and RNA-seq expression analysis. First, we wanted to verify that the iPS cells that were differentiated into dopaminergic neurons resembled dopaminergic neurons from the brain development. So we looked at development of dopaminergic neurons in the mouse embryo ventral midbrain, and we selected important markers for regional specification like EN1 and EN2, floor plate specific marker FOXA2, and dopaminergic neuron-specific markers tyrosine hydroxylase and EN1. Here in this panel, we saw QRT-PCR data of the cells from day 0 to 27 days of differentiation. And first, we verified that the cells had decreased expression of pluripotency marker OCT4 during this developmental time, and at the same time, expression of FOXA2, which is the floor plate specific marker, increased significantly in the cells. Also, EN1 and TH positive cells were detected in high levels at day 27 of differentiation in these cell populations. Then we did the FACS cell sorting, and here is the sort protocol and overview of that method, because this is really important in order to enrich and purify heterogeneous iPS cell-derived neuronal cell populations. First, we use ACTase dissociation for the 2D cultures. Then we stained the cells with NCAM positive antibody that was labeled with fluorochrome, APC. And then we did this FACS sorting with FACS area with 100 micrometer nozzle and low sheet pressure to ensure that the dopaminergic neurons were viable and didn't go through much stress during the cell sorting. We also added ROCK inhibitor to the media after cell sorting, and after 24 to 48 hours of sorting, we removed the ROCK inhibitor from the media by a partial media changing. And at day 27, before the cell sorting, we also noticed that the duplication cells, showing here in blue, had significantly lower expression of this NCAM marker compared to deletion populations. But after the cell sorting, when we differentiated cells further, up to 60 days of differentiation, when we quantified Th and 2G positive dopaminergic neurons, we didn't detect any significant differences between different genotypes in the cell population. And we detected high, over 75% positivity for Th and 2G1 expression in these cell populations to verify that these are really dopaminergic neurons. And then we did the transcriptional gene expression profiling around day 60 of the differentiation. And first we show in the panel A that the Th expression was comparable between all different genotypes studied. Then we verified that these dopaminergic neurons carried the copy number variation of 16p11.2. Showing here in red in the 16p deletion cells, these genes were downregulated significantly in these populations compared to control population. Then we did a weighted gene co-expression network analysis, which revealed modules that are correlated with the 16p gene region. And interestingly, we found out two different modules. Module 19, where the 16p deletion cells had significantly reduced expression of these genes, and at the same time, these same genes were increased in the duplication population. In module 25, we detected genes that were significantly increased in 16p deletion cells and significantly decreased in the 16p duplication cell population, shown here in blue. So this was really interesting models. And to say a couple of words about the genes in these models, we did a gene ontologic analysis, and in module 19, which contained the genes that were downregulated in the 16p deletion cells, we detected that there was genes associated to cytoplasmic vesicle part, locomotion, programmed cell death, cellular response to stress, scaffold protein binding, and ribonucleotide metabolic processes, which was really interesting finding. Then in the module 25, these are the genes that were increased in the 16p deletion cells. We detected increased expression of synaptic markers, neuron projection, cell junction, ion channel activity, neuronal cell body development, and neural differentiation. This indicates that maybe there is some deficits that can be detected in the synaptic development of these cells. And to verify the transcriptional gene expression changes, we wanted to look at the protein level, the synaptic proteins in these cells. So we stained the cells with synapsin 1 and PSD95. And we detected that in the 16p deletion cells, comparable to the transcriptional gene expression data, the markers were significantly increased in the deletion population compared to control cells, showing here in red. Also, in the Th positive dopaminergic neurons, synaptophysin expression was significantly increased in the 16p deletion population compared to control cells. Then we wanted to ask that, will this synaptic marker expression changes affect the functionality of the neurons? So first we did a patch clamping experiment where we recorded spontaneous excitatory postsynaptic currents, and we detected that the frequency of these EPCs was significantly increased in the 16p deletion cells, shown here in red in the panel F, compared to control population. To look into more detail at the patch clamping data, we detected that dopaminergic neurons with 16p deletion were hyperexcitable. So this means that when increasing current steps were injected to the cells, the number of action potentials was significantly higher in the 16p deletion cells compared to control or duplication populations, shown here in red. Then we asked that, will this affect the network level functionality of the cells? And to study this, we used two different platforms. from MaxWell Biosystems, the high-density MEA, and from Axion Biosystems, the low-density MEA. In both of these plates, we plated 100,025 sorted dopaminergic neurons together with 8,000 astrocytes, and we followed the dopaminergic neuron network development over 4 weeks with these recordings. And here we see in the upper panel in the green, the 8 positive dopaminergic neurons that have been stained after 4 weeks of recording from these tips. To say a couple of words about the method, so here we first sterilized the chips with 70% ethanol, and then we washed them with DI water. We coated the chips with PI 0.1% overnight at room temperature in borate buffer, and then we washed the chips with DI water. We then coated the chips with 20 micrograms of ml laminin overnight at the hood in 37 degrees of Celsius, and then we removed the laminin and added the cells in the single droplet with 10 microliters in the center of the chip. After 1 hour of incubation, we filled the chip with the media. And with this method, we were able to create networks that we could record for over 4 weeks. And our data shows here, really interestingly, that the 16p deletion cells were really increasingly active. They had significantly higher number of fraction of active sensors compared to control or duplication populations shown here in red in the panel B. And also we quantified the fraction of synchronized sensors, and we show that the 16p deletion networks were significantly higher number of synchronized sensors compared to control or duplication population shown here in red. Here we also saw the activity maps in the panel D and synchronized sensors in the panel E. And Hanna is going to explain in more detail the methods used to analyze these data after my talk. Then we looked in more detail the number of bursts in the networks, and we detected, interestingly, that the 16p deletion cells had significantly increased number of bursts at day 20 of differentiation compared to control or duplication cell population in the panel A. And in the panel B, we show that number of spikes per burst was significantly increased in 16p deletion cells, showing here in red, at day 20 compared to control population. We also quantified burst duration in these networks, but we didn't detect any significant differences, although there was a trend towards increased burst duration in the 16p deletion cells compared to control population. We also quantified the time between bursts in the networks, and at day 20 and also at day 18, we were able to detect decreased time between the burst in the 16p deletion population, shown here in red in panel D, compared to control or duplication cell population. And this indicates that the 16p deletion networks are firing burst more frequently compared to control cells. And here are some raster plots to show the sensors with highest burst rate in the control, duplication, and deletion cells, and also raster plots of sensors with most spikes per burst in the control, duplication, and deletion networks, which verifies that the deletion networks are truly bursting more and have higher number of spikes per burst compared to other conditions. I would like to shortly introduce the network functional effect and also the molecular mechanism behind these deficits in the dopaminergic neurons with 16p deletion. And we were able to look one of the proteins, KCD13, which is expressed in the 16p 11.2 region. This protein forms a complex with choline-3 ubiquitination complex that degrades protein called RhoA. And in the 16p deletion cells, when the KCD13 expression is decreased, RhoA expression is significantly increased in these cells. This causes stress fiber formation, spine shrinkage, axon growth inhibition, enhanced cell spreading, loss of dendritic spines, and neurite retraction, which caused macrocephalic brain growth in the patient with 16p deletion, showing here in the red. Interestingly, in the 16p duplication cells and patients, opposite deficits have been detected. So we wanted to verify, can we detect similar kind of molecular mechanism in the dopaminergic neurons in our cell model? So we first looked at the expression of KCD13 in the dopaminergic neurons, and we detected significantly reduced expression of KCD13 in the 16p deletion cells compared to control population. At the same time, we detected increased RhoA expression in the 16p deletion cells compared to control population. Then we wanted to look that could we rescue the network function by blocking the RhoA activation with specific molecule rosin, which is a specific RhoA pathway inhibitor. And we treated the cells with vehicle DMSO or rosin 1 micromolar starting from 5 days after plating in the MEA until day 60. And we did this experiment with the low density MEA that has multi-well format, 48 wells, and we were able to record the network activity in parallel from vehicle and rosin treated cells. First, we wanted to confirm that in this platform we could detect similar kind of phenotype that we see in the high density MEA recordings, and indeed we were able to show that the 16p deletion networks had significantly increased weighted mean firing rate at day 24 and day 28 after plating on the low density MEA shown here in red compared to control or duplication populations. We also quantified the number of spikes in these networks, and we were able to show that the rosin treatment reduced significantly the number of spikes in the 16p deletion networks, shown here in pink, compared to the red population, shown here in the middle. And also, we were able to verify that the number of bursts could be reduced in the 16p deletion network after treatment with rosin at day 24 of differentiation. So I'd like to summarize my talk. So today I have been telling you about the dopaminergic neurons that carry 16p 11.2 copy number variations that are hyperexcitable and have increased network activity and synchronization when they carry the 16p deletion. And also these 16p deletion cells have increased bursting phenotype, and they express increased level of RhoA. And rosin, which is a specific RhoA pathway inhibitor, can rescue the network activity in the 16p deletion dopaminergic neurons. So I'd like to thank you for your attention, and I would like to thank everybody involved in this project, our group leader, Mustafa Sahin, our collaborators from Boston Children's Hospital, MIT, and MGH. Also our lab members. I would like to thank our Human Neuron Core, Flow Cytometric Core, Translational Neuroscience Center, and MaxWell Biosystems. Thank you so much.
Thank you so much, Maria, for the wonderful talk. Yeah, it's really amazing. So for every one of you, if you have questions to Maria, feel free to add them to the chat now, or we'll also have our Q&A later after all of the presentations. So see you later, Maria. Now I'd like to welcome Hannah. Hello, Hannah. Yes.
Hello.
Hi. Welcome, and yeah, go ahead.
Okay, thank you, Marie. So I'm Hannah Pinson. I'm a co-author of the study that Maria just presented. So thank you, Maria, for the nice presentation. I have mainly been doing the data analysis of the MEA data. So I would like to give you a short introduction to the data analysis pipeline. Okay. All right. Okay, so the data analysis pipeline consists of a number of steps, of course, starting with the recording. So the recording consisted for this application of different blocks, different smaller blocks of the array that we shifted, and so we could make a recording of the whole array. Then we did a detection and removal of glitches and noise, application of bandpass filter, the spike detection, of course, and these are all steps that you can do with the MEA software. And then we developed a custom piece of code for the detection of synchronized sensors and fine-grained burst detection, because these were important properties to compare the deletion, duplication, and control cultures over time. This is an example recording where the spikes have already been detected, of course. You can see here the raster plot of the spikes over time of one of these recording of a small area of the sensor array. And so you can see that all the different sensors can have different dynamics, from almost continuous spiking to short very short bursts of activity. And the questions we just asked were, are there any synchronized groups of sensors? And is there any bursting? And of course, to which degrees are these things happening? So we have the synchronized sensors here. Loosely speaking, synchronized sensors are, of course, about which of the sensors fire together. And for this, we use the cross-correlation between spike time patterns at different sensors, which we first binned at 1 millisecond. And then a synchronized pair of sensors was a pair of sensors for which the cross-correlation between the spike time patterns exceeded a certain threshold. And then a synchronized sensor, of course, is a sensor that is a member of at least one synchronized pair. Now, this threshold definition gives you the ability to actually see more and more fine-grained structures in your network. So if you put the threshold at 0, this is basically the same as saying, okay, which are the sensors that have been spiking at least once within a millisecond during the whole recording. And if we do that for this example recording, we see, for example, 2 large structures. If we increase the threshold to 0.25, you can already see more fine-grained structures. And if you then put the threshold to much higher, to 0.75, only the very tightly synchronized regions remain. And in the paper, we used the 0.25 threshold. This allowed us to make a compiled map of all the synchronized sensors. So this consists of all these different local recordings of where we determined the synchronized regions, and all these regions got a color to give an overview of the synchronization of, or at least a local synchronization of the whole array, because we never simultaneously recorded distant parts of the array. We couldn't analyze it in this case the global synchronization. So this is about local synchronization. And then we're talking about bursting. So the bursting, or the general thing with these stem cell derived neurons, is that they exhibit highly varying dynamics, both over time, so when the cultures are developing from day 1 to 2 weeks later, or between the different types of cultures, so control, duplication, and deletion. They all have very different dynamics, and so it was a little bit difficult to use a predefined criteria for bursting, or at least we wanted to have like a more fine-grained analysis of the bursting to make sure we didn't miss anything. So for this, we used an algorithm that was developed before by Kapuku, from the paper Burst Analysis Tool for developing neuronal networks exhibiting highly varying action potential dynamics. And the advantage of this algorithm was that it could be used. So for each different spike time series, the criteria for bursting, so the thresholds basically for the detection, are set based on the statistics of this spike time series itself. So the interspike interval distribution, which make sure that the detection thresholds are based on the actual data. And I wanted also to say that it allowed us to pull together the sensors, for example, as is shown here with the highest burst rate, from across the whole array and put them together in one plot to be able to compare visually, but also, of course, in statistics between the duplication control and deletion cultures. And then finally, if you want to use the code in your own project, you can find this on GitHub. It has a number of features, so mainly the synchronized regions and the bursting algorithms. We also added a lot of plots to help us with our analysis, so that might help you as well. And there is a parallel implementation because the amount of data you generate is very high, so parallel implementation helps you to speed up the computation. And then I would like to thank Maria, of course, and all our collaborators, and of course, MaxWell Biosystems for giving us the opportunity to present our work. Thank you.
Thank you so much, Hannah. Really nice talk. And congratulations to you both, Maria and Hannah, for this wonderful paper. And yeah, we are looking forward to see what's next in this research. So now let's go to our question and answer. Once again, please feel free to add your questions here in the chat. All right, so let me welcome again both Maria and Hannah. Yes, you're here. Hello. And now we'll go to the Q&A. So first we have this question. I think this is for Maria, but Hannah, you can also answer if you're involved in this part. So a question from Shan Wang. If you already collected data from high density MEA, why still do this in the low density MEA?
Yes, that's a really good question. Thank you so much for asking this. So we selected to do low density MEA in parallel with high density MEA because we wanted to treat the cells with the drugs like rosin and vehicle in parallel. And because we were recording from 3 lines with 2 treatments. So the 48 well format from the Axiom Biosystems low density MEA allowed us to do this drug treatment experiment pretty easily. That is why we used that platform together with high density MEA.
Thank you so much, Maria. And I also would just like to mention that during that time, we still did not have the Max2 system yet, but now we have the multi-well version. So hopefully there'll be more compound application experiments being done on the Max2 system. All right. So next question. I think this is for Hannah. In what language was the implementation of the scripts that you did?
Oh, that was MATLAB, because I had the impression that the most neuroscience code is in MATLAB. So yeah, with MATLAB.
Yes, and we do also, together with the Max1 system, we do also provide a MATLAB toolbox as a starting point where you can then develop your own scripts using these functions as well. Next, we have another question. Have you characterized the subtype of your dopaminergic neurons? So this is for Maria.
Yes, that's also a really good question. So we were looking at different markers, and in our paper, in the supplemental materials, you can find a supplemental figure 3, where we have looked different markers like Neur1, and also we looked neural precursor cell markers and other neuronal subtypes to confirm that our populations were truly dopaminergic neurons. And also we characterized the cells with the dopamine transporter DAT and FOXA2. And also we did QRT-PCR with EN1. And also our transcriptional gene expression profile, we compared our dopaminergic neurons to previously published methods, and we were able to identify that our populations had floor-plate-specific markers and precursor cells, and also mature dopaminergic neurons that express dopaminergic markers. But more in detail, you can find in the supplemental figure 3 of our paper.
Thank you so much. We have another question for Maria. How do you comment on the 16p duplication results? The overall changes were much milder than the 16p deletion, but do you think there is still a clear detectable pathophysiology in the 16p duplication?
Yes, that's a really interesting question too. So we were actually surprised that we couldn't detect any bigger or more significant differences in the duplication cells compared to deletion or control populations. But we were able to look in the path clamping, for example, there is mild hypoexcitability in the 16p duplication cells compared to controls. And also we were looking the different gene expressions, and we were able to verify like genes related to dopamine signaling and drug abuse that were upregulated in the 16p duplication cells. So that were the differences that we were able to identify on those cells. But also I think in cell culture, sometimes there is a compensatory mechanism, like when there is a deletion of the cell region that is more severe phenotype than in the duplication, because the cells can control in the overexpression like internally. But when there is a loss of gene region, that is always more severe.
Thank you so much. So in all the cell cultures that you have recorded, both on the high density and the low density, you have included astrocytes with the dopaminergic neurons. So did you also try without astrocytes, and was there any significant difference between with and without astrocytes?
Well, that's a really good question too, but we actually used astrocytes in all our functional assays, because astrocytes promote the neurite development and synaptic development and the network formation in general. So the astrocytes are really crucial for the functionality of the networks. And we do think that for long-term cultures, it is beneficial to always add the astrocytes there, because also astrocytes are present in the brain. So it is, I think it's more natural to have the supportive layer of them there. And I don't think that the cells would survive or be as active without astrocytes. We haven't tested that.
Yeah. Okay. Thank you very much. I think this is also an observation by the stem cell community that culturing neurons with astrocytes really promote the network formation. So the next question is for Hannah. So we do see in some of the network activity, like what I have shown, that there are sometimes that the bursts are happening in all of the channels, all of the neurons at the same time. But in the results here in this paper, you also see some local synchronous activity and local bursts. So maybe you can elaborate a little bit more on the way that you have characterized these different localized activity. Like, for example, did you identify multiple patterns in one, and you just got the mean of that per well in terms of analysis?
I think, yeah, in visually, let's say, we always had these different overviews that allowed us to actually see that there were smaller or bigger or fine-grained structures, but to actually contribute to the statistics, to compare the different cultures, we used more average values. We also used certain thresholds to say that some sensors had to have a certain number of bursts to actually be included in the analysis, for it to have like a more like honest comparison between all of them. I think you could go further with the analysis to make it much more fine-grained with the techniques that we implemented already. But for the paper, it was just, I think, a good start and a decent analysis to just do the averages between the different cultures.
Thank you so much. So next question. I think this is more for me, Maxwell. Can the Maxwell systems also record low frequency 1 to 50 hertz local field potential signals, like corresponding to EEG frequency ranges? And yes, we can do that. The software, you can adjust the software which frequency range you would like to record, whether including the low frequency, like from 1 hertz onwards, or just the high frequency range from 300 hertz onwards. So the answer here is yes. Next question that we have, I think this is for Maria. For the astrocytes added to the assays, did they have the same genetic background as the neurons?
Yes. So the astrocytes were actually purchased from Ncardia. So they are human iPS cell-derived astrocytes that are commercially available. They don't carry the 16p11.2 copy number variations. So all the populations have a healthy background astrocytes, because we wanted to specifically focus on the dopaminergic neurons that carry the copy number variations, not the astrocytes.
Thank you so much. So yes, I think that's most of the questions. Are there any other questions from our audience? All right, I think it's all good. Thank you so much again, Maria and Hannah, for the wonderful paper and also the presentation today, and for answering the questions of our audience. And looking forward to see you again soon, and congratulations. So yeah.
Thank you.
Thank you. And
Thank you.
Yes. And for our audience, please, if you have any questions, or if you would like to contact us directly and have a one-on-one presentation together with any of our application scientists, you can contact us in info@mxwbio.com, or you can also call us via Calendly. So that's calendly.com/mxw/call. So thank you so much, and see you next time. Bye.
Bye.
Bye.